my name's Randi Johnson. I'm an assistant professor at the University of Colorado, and I just completed, I was a part of the second cohort of BioData Catalyst Fellows and just completed my fellowship in September. Next slide, please. So this slide gives a brief overview of kind of the methods that we use. The title of my fellowship was Prediction and Characterization of Genetically Regulated Expression of Target Tissues in Asthma, which is a bit of a mouthful. So the goal is that we're trying to integrate genetic data with gene expression to build prediction models that we can then use to apply in external data sets in order to predict or impute gene expression from genetic data. So as a part of our fellowship, we took publicly available scripts to do these predict scan family of methods and wrapped them, created new tools on the 7 Bridges platform of the ecosystem. So if you look at the diagrams on the bottom, the first workflow, we took gene expression data from CD4 positive T cells from the BAG study, which stands for the Barbados Asthma Genetics Study. And we ran elastic net models to predict each transcript or each expression, each gene from the genotype data that was available on the same subjects. And that's a workflow that was put together. We adjusted for certain covariates. And then saved the weights on each SNP that were informative for predicting gene expression, and then we applied those to external data sets. So that is our second workflow, which is called Predict Scan. So we use that prediction database, and we took whole genome sequencing from the additional subjects from the BAG study, so another 800 subjects, and we predicted from the gene expression or from the whole genome sequencing data levels of CD4 positive T cell gene expression, and then tested for the association between each of those predicted values and asthma. We also had worked on replicating those associations by predicting CD4 positive T cell gene expression from the CAPA study, which stands for the Consortium on Asthma among African ancestry populations in the Americas. And we've still been the last couple of months trying to refine some of those replication study or replication associations. Next slide, please. So just to highlight, the Predict Scan tools and sort of scripts are publicly available on GitHub already. And so we created tools and workflows to accomplish those, to wrap those scripts into workflows that can be used on 7 Bridges. And so on the left, at the top, you'll see the sort of diagram of each of the tools that we created, and which are the teal circles, and how they connect to one another, and data flows through that workflow to get, in the end, a gene expression database, prediction database. And then at the bottom is the second workflow that we created, which is to take the inputs, which are the gray circles, and to predict and then test for association, which are the 2 tools that are the teal circles. So both of these we've made available on DocStore. And I think for new users, one thing that's great about DocStore is that if you go there, you can find these tools. And I've highlighted on the bottom right in red the button that you can click that would open these tools into your project on 7 Bridges if you wanted to use these for your own research. Next slide, please. So this is a kind of overall summary of our discovery associations that we looked at, or sort of what we found scientifically from this work, and that was the associations between predicted CD4 positive T cell gene expression with asthma, associations with asthma from the BAG study, the whole genome sequencing. So this was conducted in 824 subjects, and we adjusted these, this is a family-based study, so we adjusted these models for kinship, for sex, for age, and then tested this association with asthma. So on the left, the histogram is showing the p-value for the associations. We tested around 4,000 genes, the predicted expression of around 4,000 genes. You can see it's a little messy. The histogram of p-values does not have quite the clear signal on the left side that we would hope to see. But on the right side, you can see each of the dots, which is one of the genes, the predicted gene expression. Along the x-axis, it's effect size, so association with asthma, and along the vertical axis, the p-value from each model. And so when we looked at a FDR, multiple comparison correction level of 0.1, there are 5 predicted gene expression in this study that were associated, at least suggestively, with asthma. And when we compare to the 155 asthma candidates, genes that have been previously identified, all 5 of these are novel. So potentially we have some interesting signal that we're following up on in the replication in the Kappa study. Next slide. So to kind of highlight the things that we used the BioData Catalyst powered by 7 bridges. So we uploaded our own data from internal studies, genotyping data, gene expression data. We also accessed the whole genome sequencing data that was funded and generated through TopMed funding and was publicly deposited in dbGaP. So we had permissions to directly access that and bring it into the system through that great connection, which I do highly recommend. If you're working with publicly available data, the ecosystem really accelerates your access to those data if you have the permissions already. The other component that we mostly used was DocStore, and so I highlighted where we've stored our workflows, and hopefully those could be useful to other people working in this space on the ecosystem. What helped us accomplish our research, so I had a graduate student who did a lot of the work with CWL. Neither of us are programmers by nature, and so we mostly work in statistical languages, not in CWL. And so asking questions of other users who were working on 7 bridges was very helpful. There are many people who are ahead of us and were quite helpful in basic things that as we sort of got past barriers and encountered common barriers that other people had already solved. The other is really, I think, don't be afraid to seek support from the platform. So the question mark button that's at the bottom of every page on 7 bridges, they probably know me by now, but we often as early users, I think I would suggest not to assume that it's something that you're doing, because sometimes there are still new glitches that haven't been identified that need to be looked into behind the scenes. And so to make people aware of that, and if it is something that's already been solved, the help desk people answering your questions will just point you to the place you need to be if it's something that you could find in the documentation. Next slide. So I think for someone wanting to accomplish a project like ours, where you're building new tools in particular, I think familiarity with Docker images, Docker files, DocStore itself would be very useful. Having someone in your group who already is familiar with CWL, I think would accelerate the development of new tools and workflows significantly, because that was and sometimes still is a barrier for us that we often have to seek support for. I know Dave's on the call, so shout out to Dave. And then I think the other is to sort of how do you think about the timeline of your project? And so I think the first tool or app that you try to create yourself, you should budget a lot more time than you would if you were doing it locally. And so if there is some particular program or set of scripts that your team uses often, it's probably worth the investment to develop it into a tool or a workflow on the ecosystem if you're going to use it many times again. So for us, we're going to be doing more prediction using these tools. And so though it took a while to get the first one or two developed, the ones that are coming after that we're kind of developing from scratch are going a lot faster. So I think in terms of troubleshooting, it's very difficult to build custom tools and app or to troubleshoot them on the ecosystem. And so I would recommend first ensuring that locally your scripts are running as expected. And then you can expect a second tool or app development to take less time, to be less of a barrier to progress. And then I think if something we have found helpful is to take existing tools. If there's one that sort of you can use as a template to edit code to work in the way that you want, but not to start from scratch, I think that also can accelerate custom development of tools and apps.